Good morning everyone, onshore and online. This is Schmidt Ocean Institute, ROV Falkor and ROV SuBastian. And this is the third dive of the northern depths of the Great Barrier Reef expedition. And we've just quickly turned on our live stream as we descend towards the seafloor so that you can see this vision that we're all watching now of this Dumbo octopus. We've been watching this girl, I think, for a little while now as we make our descent towards the seafloor. Some very interesting behaviour going on here. It appears as though the Dumbo octopus has got her tentacles curled around and is protecting something on the inside there, and we think that it might be a brood of baby Dumbo octopus. There's a lot of what looks like a lot of extra tentacles in there. So we're not entirely sure. So if anyone online is a Dumbo octopus expert, and it looks like we can see another eye in there. This is the amazing high resolution video imagery that we're capturing from ROV SuBastian as our pilot Cody zooms in there. It's quite extraordinary. So we're about 40 or 50 metres off the bottom of the sea floor at the moment at our target dive site, and fish in the background. And we were going to go live once we reached the bottom, but we decided that we needed to share this with you all as we were watching it live. My name's Marty McNeil. I'm a marine geoscientist at Queensland University of Technology in Brisbane, Australia. And today we're exploring a submarine canyon on the outer edge of the Great Barrier Reef, Queensland, Australia. And we're a little bit south of Cairns, and this canyon is known as Noggin Canyon in the northern part of the Central Great Barrier Reef. And on today's dive, we're going to descend down to about 950 metres depth at the bottom of the canyon seafloor and take a little look around there. And then we're going to make our ascent transiting up one of the side walls of the canyon. Are they moving in there? Oh, this is just amazing. Oh, here we go. Oh, look at that. What? This is just amazing. So if you've just joined us, what we're looking at here is a Dumbo octopus, and we've been following her for quite a while on our descent, and it looks as though she is holding on to a brood of babies. And we can zoom in there. There's an extraordinary amount of tentacles in there. We can see through, she's quite translucent, so we can get a bit of a glimpse through her body to what she's clutching on and the inside of those tentacles. So that's what we're looking at the moment as we make our descent towards the seafloor, where we'll start our transect for this dive. We've got 3 scientists on board ROV Falkor at the moment, myself and Dr Luke Noda also from Queensland University of Technology in Brisbane, who's a marine geologist and coral reef geology specialist. And we also have Vicky Lowe from the University of Queensland, who is a paleoceanographer. And so the 3 of us are the scientists on board right now, but we're joined remotely onshore by a whole team of science collaborators who are involved in different aspects of this expedition, ranging from geology, ecology, biology, taxonomy. Of what we're really targeting on this cruise are deep cold water sponges and corals, and of course the geology and substrates, rocks and sediments that make up the substrate and the basis of these benthic habitats. We also have some collaborators from Geoscience Australia in Canberra onshore that are joining us remotely. And the dive that we're doing today is a canyon that we visited on our previous expedition and attempted to dive here, but we weren't able to because surface conditions and the strength of the currents in this area just made it too difficult for us to put the ROV SuBastian down here. So we had to abandon that dive on the previous voyage, but we are very, very thankful to have the opportunity to come back here again and try again. The conditions here today are a little bit better than they were on the last visit, so we're able to put SuBastian down, and here's a fish. We have a couple of fish specialists from CSIRO that are collaborating on this voyage with us as well, so we'll be able to get some fish IDs from them, and of course from all of you online that are joining us on the Facebook and YouTube live feeds. Thank you for tuning in. We love to see your comments and conversations coming through. We're about 25 metres off the seafloor at the moment, so we'll just slowly make our way down. This ROV Dive's part of the first leg of our expedition to the northern depths of the Great Barrier Reef, led by our Chief Scientist Dr Robin Beaman from James Cook University, who's currently onshore, joining us remotely from Cairns. And tomorrow we're going to call into Cairns and pick up 3 more scientists, including Dr Beaman. And then we'll make our way on leg 2 of this expedition, which will take us to the far northern reaches off Cape York Peninsula, right on the outside edge of the Great Barrier Reef, where we'll be exploring the deep reef front and deep seascapes. We can see the bottom coming into view now. That I'm just momentarily casting my eye down various chats, both online and here on the ship, to see if anyone's going to tell us what some of these fish are that we're looking at. And here we have the seafloor coming into view right now, and there's a few squid around. Xenophyophore. Yes, please. So those round ball looking, spherical looking objects that you can see on the seafloor at the moment, we've seen these before on previous dives. They're called Xenophyophore, and that is a type of benthic foraminifera, and that spherical object that we're looking at is the structure that the foram builds. We've collected a couple on the previous voyage, and I think maybe in Western Australia prior to that. So we're just going to take a moment to adjust our position here and set up the cameras, and then we're going to take a couple of sediment samples here using our push core, and we'll try and pick up one or two of these Xenophyophore. Not entirely sure if the sort of that spherical structure that we see is discarded, and that the living foram has abandoned that and left it behind, or if that's still part of, somewhere in there, there'll be a single-celled, what we call a protist, a single-celled organism. If you imagine it being something a little bit like an amoeba, and foraminifera come in a huge diversity of species that are, some live on a benthic and live on the seafloor, some are planktic, they live in the water column. Many of them build a calcium carbonate, what we call a test, which is like a skeleton on their outside. These Xenophyophores are quite curious, and I really don't know too much more about them. Other than these structures that they build are incredibly complex and incredibly delicate. So it's quite a delicate operation to try and collect these things and bring them up intact. We're joined by a nice little shrimp friend swimming around there.
Smaller ones are best for fixation.
Yeah. So hi to our regular viewers on YouTube. It's nice to see you joining us again. I'm not sure what time of the day or evening it is for you where you are, but I don't think you really mind.
They're definitely agglutinating.
For us it's 10:15 in the morning and it's a lovely day outside, a little bit windy, a little bit of swell, but things could certainly be a lot worse than what they are right now. Are you going to go for that big one? Is it too big?
Get a smaller one. No,
I'm going to try a smaller one.
Do you want the smaller one or do you want the big one?
We'll do 2 push boards, so if you manage to get one in each push board, that would be delightful. The smaller one will be easier for us to try and fix.
Okay.
The larger one, of course, is more impressive to look at.
just a push sediment.
It'll be PC1, push board 1, depending on which number you guys grabbed it from. Sorry, did you guys grab number 1? Push board 1?
Yes.
Yeah, yes. This is quite a delicate operation, but our pilots, Cody and J-Rod, have done this a few times before. So the structure that the xenophyphore makes, in this case, it's agglutinated. It's rather than being any kind of mineral calcium carbonate structure, it just uses the sediment and kind of, I guess, sticks it all together with its own mucus. So that's why these things are so fragile.
Nice core.
There we go, very nice intact core. So that tells us that the sediment is very fine grained. It's holding together nicely. When it's coarser, it tends to all kind of fall apart and we have to quickly scoop it and put it into the holder. So that is very nice. Thank you very much, J-Rod and Cody. One more, please. So we're interested in these submarine canyons that are all along the edge of the Great Barrier Reef here. The head of the canyon, in this case the canyon that we're in now, goes right to the top, extends right to the top of the continental shelf and cuts in to the edge of the shelf between a couple of the coral reefs that are up on the shallow shelf up there. It's what we call a shelf incised canyon, and it is shelf connected, and that means that there is not a coral reef in the way blocking the head of the canyon. And so what this means is that the submarine canyon is a conduit, it's a transport pathway for sediments coming down from on land all the way across the shelf of the Great Barrier Reef Lagoon, and all of the sediment that is shed from the coral reefs on the way, and ends up getting flushed down these submarine canyons and eventually washed down into the deep ocean basin. So it's interesting for us to collect these sediment samples and look at the sidewalls of the canyons. It gives us an idea of sediment transport over geological time. But in addition to that, of course, all of these places are habitat. So we're interested in the habitat potential for the different types of canyons. So we've been surveying different structured canyons, blind canyons, which are what we call slope confined, a variety of different geomorphologies and what we'll do onshore later on with our colleagues from Geoscience Australia is that they'll be assessing the differences between these different types of submarine canyons and seeing whether the ecological communities are the same or different between these different canyon types. And that's fantastic. That's our second xenophyre collection. These look to be the same as the same type of xenophyre that we collected on the previous expedition. And we'll have to be very, very careful with these when we bring them up on deck at the end of the dive. They're very, very delicate. There's really not anything holding them together except for mucus. Wonderful. Fantastic.
That's good on course.
Yeah, good to get on course. So we've planned a transect today that's about 1,000 metres in length, and we'll make our way across the canyon axis to one sidewall, and then we'll go across and up the other opposite sidewall where we'll make our ascent up towards, we'll be ending the dive in about 700 metres of water depth. So we won't go all the way to the top of the canyon today. The time that we have available, that will be plenty of ground for us to cover. When we get to the walls of the canyon, we'll be looking to see whether we have any rock outcrop, some hard substrates or any rocky boulders that have come down, and those hard substrates will then become something that cold water corals and sponges can attach to, so we'll be having a close look at those. Looks like we've got a sea cucumber there. There we go. Swimming holothurian, swimming sea cucumber. A few of them there. And there's lots going on here.
Sorry.
It's okay.
Just
Interesting looking shrimp slash something there. Pausing for a moment on my internal chat here that we're talking to all our collaborators with, just to let them know what we're seeing here, get their attention. We have what looks like a whip coral or sea pen here. We might just take a close look at that. We do our usual tentacle count. I think this is a soft coral, which is 2, 3, 4, 8 tentacles. Thank you, Cody. So our targets today are not these guys, but it's great for us to collect vision of these sorts of things for others that will be wanting to look at this later on. Our sampling targets today, as well as some geological samples, are deep cold water sponges that we'll be collecting for the Queensland Museum, and deep cold hard corals and black corals that we'll be collecting for collaborators from the Museum of Tropical Queensland. And if you've been tuning into our dives regularly, you'll know that our colleague Jeremy Horowitz, who's from James Cook University in Townsville, has a particular interest in the black corals. And we'll be collecting some for him today, hopefully. So Jeremy wasn't able to join us on board and sail with us on this expedition, but he's certainly joining us remotely. So where we're situated at the moment is what we call the canyon axis. So if you imagine submarine canyon, something like the Grand Canyon, although where we are right now it's not quite on that scale, and we're at the bottom of the V of the canyon at the moment. Lots of fish around down here this morning. We're in 960 metres of water depth at the moment. It's about 4.5 degrees Celsius temperature down here. Few fish around. I'm just waiting to see if any of our fish guys are online. Nini on YouTube says we should zoom on a few fish. What's this? Nini is a fish guy. Excellent. Lots of halosaur. One suggestion online is that these are basketwork eels. Beautiful. Hi John, nice to see you online. Aldravandia halosaur with a filamentous tail. Cute, yes, that it is. A few more of these holothurians on the seafloor here, which holothurian is sea cucumber. Oh, that guy's pretty. Yeah. And a few shrimp around. Oh, yeah. Fish bites. Plenty of fish action here. Gemfish. Hi, Will. Good to see you online. Thank you for joining us and providing some fish ID. Squid ink.
Bridge. Bridge. Yep, I got you now, Tony. You got me?
So if you've just tuned in and joined us on the live stream, we're diving in the Noggin Canyon,
Hey, Tony, you got me?
a little bit south of Cairns, right on the outside edge of the Great Barrier Reef, on the edge of Australia's continental shelf. And this is a submarine canyon that connects the shallow upper part of the Great Barrier Reef, the continental shelf, and it runs down to about 1,200 metres out on the Coral Sea Basin, seaward, offshore of the Great Barrier Reef. And what we're doing today is we'll be transiting up the side wall of the canyon from about 950 metres up to about 700 metres. Along the way, on the first part of the dive, we'll be trying to do some sampling of some of the geology and biology. We're interested in sponges, hard corals and black corals. Well, we're interested in everything, but those are the things that we have permits to collect on this expedition. And then, of course, the video imagery of the entire habitat and ecosystem and communities of different organisms that are living here. And our colleagues at Geoscience Australia will use our video surveys that we will do on the second part of the dive, and they'll be doing some quantitative analysis of the different species that we find living along a specific transect, which they've been doing regularly on canyon dives in Western Australia and on the previous expedition out to the Queensland Plateau and the Coral Sea. And they'll be making comparisons of the communities that are living in these different types of canyons. So we're very happy to be able to collect that information for them, although they can't be on the ship for this expedition. It's important for us to collaborate with as many different scientists with different specialties as we can to make the most of the opportunity of having ship time on board ROV Falkor and having the amazing technology that's available to us with ROV SuBastian. And so we collaborate with as many different scientists as we can to maximise our data collection. And there's one of those holothurians, sea cucumbers, swimming away now. Oh, yeah. So we quite regularly see these guys in these water depths on this soft sediment. Very graceful. Nice little shrimp down there. thing there.
You're right there, but you're right there. Is that the way back there? Yeah. You want it back here?
We're at 950 metres water depth at the moment. It's just under 5 degrees Celsius. And we've had quite a lot of fish and shrimp action here this morning. And if you missed the very start of the live stream, we had some amazing vision of a Dumbo octopus while we were making our descent towards the seabed. So we quickly went live so that those of you watching at home on shore could see that with us. It was very interesting. The Dumbo octopus had its tentacles curled right in and wasn't letting go, so we were curious about what was going on, and it appeared as though she might have been clutching on to a brood of babies. We could see the tissue was quite translucent, and when we zoomed right in we could kind of see through it a bit, and there were an awful lot of tentacles in there, so it looked like she was clutching on to a brood of babies and protecting them. Certainly wasn't letting go or unfurling those tentacles like we normally see the Dumbo octopus when they're swimming around. So that was quite a cool little highlight right at the beginning of the dive. Sorry if you missed it, but you'll be able to go back and watch the video later. Of course, these all go up on YouTube, on the Schmidt Ocean Institute YouTube channel. And you can follow along. This particular expedition is called the Northern Depths of the Great Barrier Reef, and our social media hashtag is EdgeGBR. Of course, we have our cruise webpage on the Schmidt Ocean Institute website, where you can follow along with cruise updates and cruise blogs. If we see.
It's very light here this morning.
There's a lot of things happening. Thinking about this out?
This morning we've got Cody in the pilot chair and J-Rod co-piloting. Let's go in and check that out. Cup coral? Is that Flabella? We would like to collect that, please.
Okay.
Bless you. Is that an anemone or is it a cup coral?
It's got a skeleton.
It's got a skeleton? Yep. Yes, excellent.
Very thought.
Yes, collect that guy, please. Yes, a few comments on the YouTube chat. There is a fair bit of packet rustling going on here. It is snack time.
Where would you like this to go?
We could go in box 1A, please. want to talk about your interest in flabellum and what you would like to do with it? Yep, jump in there. Sorry, did you get the collection log first? Yeah, we're just going to collect this flabellum cup coral, solitary coral, and get that stowed away, and we'll log that. And then I'm going to be joined in the chair here by Dr Luke Notiff from Queensland University of Technology. This sample is of particular interest to him, and he'll come and tell us a little bit about this particular type of solitary coral and what our scientific interest is on this particular group.
Keep it moving.
Yep, keep moving, thank you. Go for it.
Hi everybody, this is Dr Luke Notiff from Queensland University of Technology. So one of the things that I work on is how corals make their skeletons, and I have a PhD student, Brett Lewis, who's working on aspects of that in shallow corals, but we're particularly interested in this deep coral flabellum because it has a fairly unique skeletal structure in that it has an outer wall. Most of the modern hard corals on the reefs are colonial, so have many different polyps and many different animals within the one colony. So they generally don't have, there's very few ones that are solitary. So there's the fungidae and some of the cup corals that are solitary, but this particular one has a nice wall called a theca on the outer part of the solitary polyp, and we're particularly interested in how that forms, with some reference to how some of those colonial corals do it, because they have to do it in a very different way, because there's many different polyps and they have to rearrange their tissue in quite a different fashion. So this is kind of our type sample to compare to. So it's fortuitous that we came across one. I've been looking for one of these for years actually, and we just happen to have all of the equipment ready to go. So just a nice clear looking sea pen there, I think. If Jeremy's online.
Not sure if he is.
No? So
Probably not one of his collection targets.
Go.
Yep, keep going.
Roger.
Yep, thank you.
interesting joined xenophyophore there. How do they reproduce?
What, the xenophyophores?
Mm.
I have no idea.
Mm.
No, you're right, stay there.
Text Brett.
Bear with us for a moment, we're just shaking a bit of sediment off Sebastian. And then we'll get back into our position and continue along our transit towards the wall of the canyon. So the sediment in the push cores that we collected earlier on at the beginning of the dive when we came down to the seafloor, we will take those samples back to our lab back on shore at the end of the expedition. And we'll be going through, well, first of all, we'll gently try and get the xenophyphore off the top of the core and try and keep that intact. And then we'll be looking at those sediment samples, and first of all, we'll analyse all of that under the microscope and have a look at what the sediment is comprised of. And that will give us an idea of where it has come from. If there are any microfossils in there, we can make an attempt to try and date those. And then that will give us some indication of the sedimentation rate, how long and at what, how much of the sediment has been deposited and accumulating here.
What is that?
If you can hear some talk in the background here, we're doing a ship to shore hookup at the moment with a primary school or a preschool in the United States. So this is a live ship to shore hookup with some young kids. I think they're around about 5 or 6 year olds, and they're joining us live from their classroom, just as we're joining you live on YouTube and Facebook. So we're showing them around the ship, and at the moment we've brought their camera into the control room so they can see what's going on during an ROV dive in the control room. So you can probably hear a little bit of that in the background. You can get a sense of the slope gradient now as we approach the wall of the canyon. You can see that we're going upslope a little bit now.
Too heavy.
Marty, do you want to go right up? Like that? To get to that start?
Once we get to that
Or-
start, then we have to stick onto that transect, so I don't mind if we kind of slowly make our way towards
Linger
there and look
around.
around. Because once we get on that, we have to,
Stay on that.
and we've got ages.
See on a group of me.
Yeah.
That's a great question. I am not on the ship right now. I'm here in Kailua, the same town as you guys, and I get to connect to Falkor to the ship remotely through these live connections. What's really special about Falkor and SuBastian
You can still talk. They won't. You're fine.
Thanks. So it looks like we're starting our ascent well and truly up the side of the canyon wall. Not seeing any hard rock outcrops. It's well and truly covered by a sediment drape. And it looks like we're coming over these sort of undulating gullies that we saw before on the previous expedition. If we do see any rock outcrops, we'll be certainly wanting to go and have a close look at those. What's that? Is that a squid?
Yeah, it's good.
Very nice. Very cute squid. It's like, what's that, a brittle star sitting underneath that xenophyre there? We're on quite a steep slope here, and the sediment is certainly very cohesive, so for it to settle on this steep slope and stay intact suggests that it's very fine-grained, sticky mud. Which is kind of nice for us because when we bring the cores up on deck and have to process them in the lab, they'll stick together and not fall apart and slosh everywhere. Try and get a good look at this squid.
Get gross.
Yeah. So we're just going to take the opportunity to have a look around for a little bit. This is certainly just sort of exploratory dive that we're doing. Take a look around and see what we find living down here, both on the seabed and in the water column. Can you take a look at that? And in a little while we will start what we call a benthic transect, which will be a video survey for our colleagues at Geoscience Australia. We won't be sampling once we start that transect. We'll just be flying at a consistent speed and altitude all the way along that 500 metre transect, and that's so that they can get a comparative data set to compare with what they've collected from other submarine canyons, both here on the east coast in the Great Barrier Reef and out in the Coral Sea, and also on the west coast of Australia. So currently we're on leg one of our northern depths of the Great Barrier Reef expedition. We left Brisbane almost a week ago now, and we've sailed up along the edge of Australia's continental shelf, to where we are now on the outside edge of the Great Barrier Reef. We did a midwater plankton survey for our colleague Dr Dougal Lindsay, who joins us from Jams Tech in Japan, from Tokyo. And this is our third dive on this expedition, and then tomorrow we're going to make a port call in Cairns. Oh, hello. Who's this guy? Wow. That is spectacular. And we'll be picking up 3 more scientists in Cairns, including this expedition's voyage chief scientist, Dr Robin Beaman from James Cook University in Cairns, and then we'll get on our way on leg 2 of this expedition, which will take us all the way up to the far northern reaches of the outside edge of the Great Barrier Reef, offshore of Cape York Peninsula, which is a very remote part of Queensland, part of northern Australia, and to be outside on the outer edge of the Great Barrier Reef is even more remote, very challenging to get to, not easy. It's a long way north from any major port, Cairns.
Got some rocks coming, Marty.
Oh, fabulous.
Or, something.
And
something coming.
got something. Okay. Oh, looks like we've got some different substrate happening here. Still looks very muddy, draped with mud, but and lots of burrows, lots of burrowing, and these little rills or gullies that are formed down there. So these little burrows will be home to something or someone. The larger ones are usually or often made by isopods, and we certainly saw some very large isopods in their burrows on the previous expedition.
Bridge, do you want me to hang out here for a moment so you don't get into a circle of death?
I'm moving to the east a little bit, but we should be alright where we are.
So I'll keep moving to the waypoint?
No, I'll move the waypoint to the east a little bit.
Oh, okay, sweet. I thought you were. Sorry, I brought you in.
Alright.
No, good. Yeah, that's cool. We'll go there.
Alright.
So once we're on that 500 metre benthic transect, we have to be very disciplined and stick to the transect and not get too distracted. I can see that there's already some comments on the YouTube chat from regular viewers who have joined us on the previous expeditions where we were doing these 500 metre transects. And it's easy to get distracted. We always say we stop for Dumbo octopus, nautilus and giant isopods. Sorry? Is it one of those worms? Sorry, we're just seeing what we're looking at here. Looks like it could be a worm. So we wondered if this could be one of these Ichthyoid worms that we've also seen on other dives, and they.
Damn, Cody, all picky.
What was that?
Keep going.
Nice brittle star there. Unless you want to give the worm a nudge and see what it does.
Yeah. You definitely can do that.
So our worm or not worm test is to give it a little tickle and see if it retracts itself.
What a room page, yeah.
Alright.
Oh, wet buddy. Yep, that was me, sorry. I forgot I had lock on.
Don't touch it too hard.
Just a gentle nudge. We are moving around a little bit here at the moment. Nope, nobody home.
Nope.
No one home.
Knock,
It's,
knock.
may have been a worm at one point. All right. So we'll just keep looking around before we lock ourselves into our 500-meter transect.
We are about 50 meters away.
We got hours. Have a look at that, please. That thing in there. I can hear you.
Sleeping?
C-pen maybe? Yep. Yes. A C-pen.
You want it?
No, we don't want to collect it. Sorry.
Let's go on the other side of this, see if there's anything.
Yeah.
I have a good feeling.
Excellent.
There's something big out there.
Mm-hmm.
Sounding, dude. I thought it was a
Nice
big
little
rock.
sea star there.
Take a look at this for the unknown explorer. Sorry, starfish.
So if you've just joined us, this is the third ROV dive of the northern depths of the Great Barrier Reef expedition, and we're diving today in the Noggin Canyon. We're just a little bit south of Cairns on a submarine canyon that is connected to the shallow part of the Great Barrier Reef on the continental shelf, and it flows all the way down the edge of the continental slope out to the basin out at, well, at the moment we're in, we started our dive in 950 metres of water, but it's the canyon foot certainly goes out deeper than that. And we're making our way up the side wall of the canyon. And we're interested to see what type of habitat this is, what type of substrate, which at the moment looks like it's very much soft sediment draped over the walls of the canyon. It's a swimming holothurian that we're looking at now. And we'll be looking at the benthic communities and anything that we see in the water column.
Not a very good swimmer.
No.
Probably just 8.
It's like see. Look like swimming endeavors.
We'll be coming up this wall of this canyon up to about 700 metres of depth towards the end of the dive.
Doesn't seem itself.
Yeah, I remember the night when I caught this stuff on the bio box. I just said, don't open up the bio box. Yeah, what's that? Not able to do. I mean, you're go about it. God, that's really, really big.
We look at, sorry Cody, there's something just down here to take a look at. Brakes on.
You want to look at?
Something just down here.
Right there.
Maybe it was that. Sorry, whatever that is or was. Sure, let's take a look. What is that?
Sea pig?
Is it a Hawthornian?
It's like sea pig.
A what? Sea pig?
Yeah,
What's a sea pig?
that thing. They had these little like antennas. No, it's a cute thing.
It's just having a moment. Captured live to the world
All in the name of science.
in the name of science. So this behaviour will be of interest to someone who specialises in deep benthic holothurians.
That's how they make the spiral.
And there you go.
So much like
That feels better. So when you see these traces on the seafloor, now you know exactly how those are made. All right, we're keeping our eye out for Jeremy's whip coral with a double row of polyps. Is that an urchin? Zoom in here, it looks like it could be a sea urchin. An irregular echinoid. Yeah, don't know if that's actually alive or just discarded. There's only one way to find out.
It's disgusting.
Yeah, but Chris is bored.
Hold it. Touch it.
No, it's okay. We're pretty sure it's
I'm
no
already
one's
out
home.
there.
Oh, okay, okay. You go ahead. You go ahead. Yep.
Hard.
It's hard. It's hard, but no one's home. What is that? Is that a gastropod? It looks like it's a gastropod. Bless you. Gastropod is a snail, a sea snail, a type of mollusk. So I don't think we'll collect this guy. Continue having a look around. And what is this? What is this? Looks like
It's tied off, so
yeah. It's anchored itself through some kind of appendage there.
We'll follow that to where it goes.
Yeah. Where is that attached to? That is, wow, that is really
I think
long.
it's gone.
What's it hanging on to?
The only small rock that's over.
There you go.
Just attached to the sediment. Wow. go back and check this guy out. Nice brittle star there. Sea star. We've been complimented on our polite manners by calling holothurian poo tubes traces. That is what we call them because usually, well, in sediment and even in rocks and fossils, you don't see the organism. All you see is a trace of the behaviour that they've left behind. So it could be a feeding trace, feeding burrows. It could be a poo trace, which is what we've just seen. It could be escape burrows. They make all sorts of different structures, and we see them in the soft sediment. And we also, they can preserve in the fossil record. And so in fossils where we don't have the soft-bodied organism, all we have to try and work out something about the ecology and the behaviour of the organism is evidence of the traces that they leave behind. So we're having a bit of a conversation here to try and figure out what this thing is. What were the thoughts?
I'm going to back and underneath that.
Yeah. Means it obviously has tentacles. Looks like, and that, yeah, what we're seeing on the top now looks like it could be the base. Scapopod. This is curious.
They look like mesenteries coming out.
Benthic siphonophore. Thank you for the contribution, Benthic siphonophore.
Ready to go?
Yeah.
Ready?
Yeah. All right, carry on our way. We need to keep our eye out for anything that looks whip-ish and check it out for Jeremy. Oh. Questions? Oh yeah. Kids always have the best questions, don't they? Oh, we could hear it in here. Yeah. What's that? Oh, there's another one of those benthic siphonophores. Yeah. Can we check that out? That. Is it anemone or an anemone or cup coral? Just an anemone? Yeah. What's at the base there? Yeah, what's this little thing here? No, not that. Up here. There.
Oh, it's one of those ones with the thing.
Oh, that's one of those crabs that wears an anemone on its shell. That's an oversized accessory. It's quite the show off. Yeah. Interesting. Okay. We'll head towards our waypoint. We won't be collecting anything. If we see something really amazing, we can log it. But for oh yeah.
You ready?
Yeah, ready. So we're going to make our way to the start of our official start point for our 500 metre benthic transect. This is for a quantitative analysis that we do, so we'll be very disciplined and we'll stick to the waypoints and we won't get too distracted. So the pilots will settle into our optimal position. We'll be trying to fly at a consistent altitude and speed so that we can make quantitative comparisons between this transect and ones that we've done in other submarine canyons on previous voyages. So we're just going to adjust our lighting and our altitude.
That shadow. No, that shadow is.
That's what we're trying to figure out. Shadow. I know what that is.
And you want more lights or less lights?
You take a push forward this time.
No.
Alright, Marty.
Yep.
Happy with that?
We're happy. You ready? Yep. Okay.
2 meters up, right?
2 meters. Yep.
Very far. That's 2 meters there.
Good. Yep.
And here we go.
Good to go. Start the transect. Yep. Excellent.
And transect.
Great. So we'll just make our way along this transect for about 500 metres. We've already had a, oh, what's that? Something that is not in the 4K science camera that you're looking at right now. It's in one of the other cameras on the ROV that the pilots can see, the situational camera. It was a siphonal that was in the water column. But we're focusing on the benthic habitat for this transect. So benthic, that means on the seafloor. So we'll continue along with our camera directed towards the seafloor.
Is that a good speed?
Yeah, that's a good speed. Thank you. And then onshore, after the cruise, another group of scientists will take this video footage and they'll be replaying it back and they will be annotating every single organism that they see in each frame, in each field of view, including the type of substrate and any other observations like that bioturbation, burrows, poo tubes, anything else that we see. And that will help them to put together a picture of these benthic communities that we're seeing on the sidewalls of these canyons. And one of the hypotheses that they'll be testing is whether different types of submarine canyons with different geomorphologies host different communities. So some of these submarine canyons incise the shelf where they meet the Great Barrier Reef. The head of the canyon cuts into the edge of the continental shelf, and we call that shelf incised and shelf connected. And other canyons don't do that. The head of the canyon is sort of partway down the continental slope. We call those blind canyons or slope confined. And there's a few other sort of subcategories within those. And different slope gradients, different aspects, different parameters, and all those parameters combined we sort of call the geomorphology, which is the sort of shapes and aspects of a particular substrate or habitat. And then that may then influence the type of community that can live there. And our colleagues from Geoscience Australia that are doing that benthic habitat work were on board ROV Falkor on the previous expedition in the Coral Sea out to the Queensland Plateau, and also on a voyage prior to that in Western Australia. They weren't able to join us for this particular voyage, but we're happy to undertake this survey for them and collect a data set that will add to their growing collection of canyon transect data sets. It's really such a rare opportunity to get ship time to be able to come to see in these remote locations, and so we try and maximise the samples that we collect and the data that we collect for as many different groups, research groups and scientists as we can. Not just the scientists that are on board Falkor, but also our colleagues and collaborators that we work with at other universities and institutions around Australia and also all around the world. Is that a whip coral? So the value of these research cruises, really, I mean, you just about couldn't quantify the value. These data sets will get continued to work on probably for decades, really, to be honest. Had some informative updates coming through on the YouTube chat. Sorry, say again, Chris? I have no idea. Meteorites. The question was what's created all these divots. So they could be bioturbation from an animal that has nestled in there and created those divots. They could also be a result of the currents that are kind of circulating through here. So any type of, well, we call them bed forms. Bed forms can be current ripples or dunes that are caused by water movement, picking up sediment grains and depositing them elsewhere, which is an entire field of research on its own. People measure these things and can work out current velocities and directions and things from the different types of bed forms.
We have gone a total of 78 meters.
28 meters. So we're about nearly 80 meters along our benthic transect. We need to do a total of 500 meters. That looks like one of those echeurid worms down there. Is that a... Just wondered whether that was a discarded nautilus shell that we just passed over there, or it might have been one of those echinoderms that we saw earlier. Once we pick up the rest of our science team from Cairns tomorrow, we'll be making our way north, northward from here for approximately 3 weeks. We'll be doing a lot of mapping of the seafloor using ROV Falkor's very excellent multi-beam sonar mapping system. Gives us very clean, high resolution imagery, well not imagery, but maps of the seafloor. There's a couple of those worms, echeurid worms. We'll be going to some really remote the outer edge of the very far northern Great Barrier Reef, where there's really been few surveys at all really, particularly compared to the shallow parts of the Great Barrier Reef. So as much as we've been studying the Great Barrier Reef for decades, and you'd think that we would just about know all there is to know about the Great Barrier Reef, but that's certainly not the case. The iconic shallow coral reefs that we picture in our mind when we think about the Great Barrier Reef actually only make up a very, very small percentage of the area of the Great Barrier Reef Marine Park and the Great Barrier Reef World Heritage Area. Most of the area is actually these deep regions that are offshore, still part of the marine park, still part of the World Heritage Area, and so it's very important that we understand these habitats, not just the seafloor but the water column as well, so that we get a much better picture of everything that we've just been talking about, really, the geology, the geomorphology, the benthic communities, the biodiversity, not just individual species but the communities that are living together, and that all contributes towards the management and protection of the marine park. We need to know what's down here in order to make management decisions and conservation decisions and ensure that we're protecting the right things in the right areas. And of course, for ourselves, there's the scientific objectives as well. So some of us are interested in the rocks and sediments, some are interested in particular species for their taxonomic work. So some of the samples of black corals and sponges that we're collecting, they'll be taken back to museums and have DNA analysis done on those to try and resolve the genetic relationships between these different families, different groups. And all of this is new work. I mean, nearly every dive that we had on the last expedition, we had a Falkor first. So it was either a new species or it was a range expansion. So not a new species, but the first time it's been observed in this particular region, in this location. So lots of new discoveries to be made. So we're really on a voyage of exploration and discovery over the next few weeks to the outer edge of the northern Great Barrier Reef. So lots of scientific studies and monitoring does of course go on in the shallow parts of the Great Barrier Reef. There are a number of locations where there are research stations, and groups of scientists go there and conduct their work based from the research station. That looks like a swimming holothurian in the top right of the picture there, going very slow. And of course the Australian Institute of Marine Science has their long-term monitoring program where they go and visit specific reefs and conduct, again, quantitative surveys sort of on a rolling cycle. I'm not sure if it's a 5-yearly cycle, correct me if I'm wrong about that. So there are places that are very well studied and are monitored, and so those are the places that we know a lot about. But the Great Barrier Reef is huge, and it's impossible to be everywhere and monitor everything. And there's certainly these remote regions and deep seascapes that we really have not had the opportunity to have certainly an ROV down, like ROV SuBastian. Some surveys have been able to put down drop cameras or towed cameras, but again, you know, the coverage is very sparse. So they're just sort of snapshots from a single location and point in time. So we've got 3 weeks ahead of us coming up on ROV Falkor, where we can map these deep seascape features and use SuBastian and get some really the first video imagery of these habitats and what's living there. So particularly in these deep reaches, just about everything that we collect has a high chance of being a new collection for the Queensland Museum and the Museum of Tropical Queensland.
What are you doing, Jody?
Ready? Grabbing his mouth over here, okay? We have gone a total of 175 point.
Outlander and Keep Grant's character and who's that over there?
Oh, did I? Never to do that one.
There's a question from, hi Jason, there's a question, who gets to count the xenophyphores? Not me. That will be one of our colleagues at Geoscience Australia that will go over this footage and get to count the xenophyphores. Certainly plenty of them. They might get a summer intern to do that job. Is that cutlass fish?
Mm-hmm.
Yeah, cutlass fish up there to the top right.
Nice fish.
Very nice fish, colourful. So we're seeing quite a few of these echeurid worms. These are these long, thin worms that have a sort of two, sort of like antennae. I'm sure they're not actually antennae, but that's what they look like. And we see these quite regularly. We've seen quite a few of these. We're certainly seeing a lot here. And they have that very long, thin tube or stem that they attach down somewhere down in their burrow. And if we touch one of those, which we won't do because we're on our transect, they retract right back into their burrow.
Switch times, but yeah, switch time. So just did waypoint ball on that second speed. I don't know if I'll work that way. Huh? Oh yeah, right. Oh hey John. Hi bud.
What's been doing, John boy?
Thanks.
Now want to hang out with us today?
Did you go eat?
No,
Which one?
I don't know.
Why?
I didn't go eat.
Why?
Because I had some
Why?
other work stuff.
Been a little busy this morning, John.
Sorry?
You've been a little busy this morning.
A little bit. That's all good. All in the name of science.
Had quite a few activities on the ship this morning, so we've all been quite busy. holothurians. I want to capture them. And for us on board the ship at the moment, it's getting on around lunchtime, so the topic of conversation is always what's on the menu today.
It smells good out there in the hallway.
It smells good in the hallway. Yesterday it was fabulous. It was the best fettuccine carbonara I think I've ever had.
Very rich, made for a sluggish afternoon.
Yeah. Yeah, it was definitely one to put you in a carb coma for the afternoon. There's some great fish ID chit chat going on the YouTube feed. To be honest, I can't keep up. By the time I get my eye in and read those comments, we're long past the particular fish in question. But we're seeing plenty of these holothurians on the seafloor here.
230 meters, almost halfway there.
Thank you, Cody. We're nearly halfway along our benthic transect. Oh, thank you. Yeah, I did start to see those comments earlier. We started talking about the Dumbo octopus and got distracted, as tends to happen. So there was some comments earlier about what that Dumbo octopus that we saw right at the start of the dive may or may not have been doing. Certainly had its tentacles retracted quite protectively, it appeared, and so not holding eggs, but no, not eggs. We could see that it appeared that as if there were way too many tentacles in there for one Dumbo octopus, and wondered whether it was actually holding on to juveniles. But certainly, yeah, holding something. No, not eggs. No, we didn't think it was eggs. It looked like they were, babies. But from what I understand, most octopus brood their eggs and then leave them. And once the eggs hatch into baby octopus, they're kind of on their own. So I'm not too sure about Dumbo octopus, what their behaviour is. But we certainly got a lot of good video of that, so we can go back and check that out later. The inset in your screen at the moment is the 3D imagery that we have of the seafloor from mapping that we've done previously. Very nice fly through here. Oh, there's a little ray. Oh, oh, feisty. Wow.
Wrapped up like a deuce.
Another runner in the night.
So what you're looking at here is some 3D vision of these submarine canyons that we've been talking about, and this gives you a good idea of what we're talking about when we talk about submarine canyons. And that white track is the ROV Dive track that we've been following today, and the little yellow box there is ROV SuBastian, not to scale. And that's our position on the side wall of this canyon as we make our way to the top of that ridge. The top there is in about 700 meters of water. And that's a good shot of Cape York Peninsula and the grey on the left, and then the coloured areas is the seafloor, colour coded. The reds and oranges. Orange is the shallow parts of the Great Barrier Reef. Those red colours are coral reefs that are either at sea level or just below sea level. So that's the flat part of the continental shelf. And then as we fly down and zoom there, we come to the shelf edge, the shelf break, which you could think of as the drop-off. and then the deep sea basin out on the right hand side is connected via these submarine canyons.
The brownish colored stuff is from last trip.
Yeah, so there's a couple of different colorways that you've got there. The brownish, yellowish brownish colouring is the mapping that we did on the previous expedition that we've overlaid on the top. And so we got some very good high resolution multi-beam coverage from the last trip, which helps us enormously with planning our ROV dives. We obviously don't want any surprises. We want to know exactly where we're going and what's down there before we put the ROV down. In terms of the substrate, certainly what's living down there is always a surprise.
No, we're trying to think of it here. Also, we can catch up on some maintenance and do some proactive improvements. Boy, watch out.
140%. Another swimming cucumber coming through.
Yep, we can just see in the situation camera, there's one of these swimming holothurians, swimming sea cucumber, going to come into view shortly. No. No. We saw it in one of the other cameras on SuBastian, but he didn't swim into view on the science cam, so on the 4K, so you guys didn't get to see it. We've got all sorts of other things happening in the water column around us, but because we're on our benthic transect, we need to keep in this position with our camera, with our 4K camera facing downwards towards the seafloor. And we're doing a very good job today of not getting distracted.
We're doing a pretty good job. Proud. Because Jeremy's not here. Right, Jeremy?
I wasn't naming any names. More than one that tends to get distracted.
I don't think Jeremy appreciated the popcorn and popcorn today. They're probably not going to be happy with that one.
Yeah.
It's funny. Okay, we're not trying to say, oh, wait.
Yeah. So we tend to get distracted by nautilus, Dumbo octopus, squid, giant. Oh. Dear, oh dear. There was a squid behind us that did not survive its encounter with ROV SuBastian. Is he right? Did he survive? Oh, okay. He inked for sure.
Good one.
I like the cold.
Yeah. Transect.
We're just doing a change of shift for the ROV pilots.
You're almost done.
As we mentioned before, it's lunchtime here on ROV Falkor, so we'll do a bit of swapping around, give everyone a chance to take a break and go and have some lunch.
How's lunch?
Fried rice.
What was for lunch today, Gerard?
Fried rice.
Fried rice? Chinese?
There's other things. Salads and whatnot.
Ah. Sounds good. Yeah, we've seen a lot of those worm thingies.
No acorns.
No, we haven't seen any acorn worms on this dive. Which
So I
is,
see cucumber here and watch it fall.
So we have been keeping our eyes out for acorn worms. On a previous expedition, J-Rod managed to collect an acorn worm and keep it intact, which was the first, what was it, first successful acorn worm collection in Australia. Because they're very, very fragile and they tend to just fall apart. And so we're keeping our eye out for any more and try and get a second collection of acorn worm. But we haven't seen any on this dive. Yep, I believe the worm did survive the trip. Not the one we tried to collect one on the previous voyage, and that one did not survive. But the first one that was collected in Western Australia, I believe, survived. We're about 3/4 of the way along our transect at the moment. Seen lots of holothurians, lots of echeurid worms, lots of sediment, bit of biotubation, trails and traces. And a lot of xenophyrophores, but we're not really seeing any where we are right here right now.
There's a question about the worms, the
Where am I
branching
seeing?
worms.
What are they? Those branching worms that we're seeing are called echeurid worms. I think they're a type of polychaete. Question mark. Worms are not my specialty, but we have seen a lot of them on previous voyages, so I don't know too much more about them other than that, but they have those two appendages at what I assume is the head end, and I assume the tail end is anchored back in a burrow, and they can get very, very long. We followed one on a previous dive on a previous expedition that was a few meters long. We weren't even entirely sure if that was what it was, but we gave it a little boop and it retracted itself back into its burrow and confirmed that it was indeed one of those echeurid worms.
Yeah, polychaetes.
Polychaetes, yep, confirmed. They are polychaete worms.
Saturday polychaetes.
And there's one coming into view right now. I'm going to step out for a minute and go and have some lunch myself. So I'm going to leave you in the capable hands of J-Rod and Chris, and the scientists in the control room at the moment are Luke and Vicky, who may or may not get on and have a chat with you, but I'm going to go and have some lunch and I'll join you back again shortly.
Hi everybody, a little squid playing in the mud. Some burrows of some description. Isopods there. So we are currently at about 710 meters depth. Water temperature is 7.2 degrees Celsius. We're on a 500 meter transect, video transect. So we're avoiding sampling at this point and stopping. And we've got about 75 meters left of this 500 meter transect.
They're used to hearing us talk about nonsense. What we eat, what we miss eating, what we plan on eating when we get home.
I've been looking at Floridian recipes lately. Like so
Chicken.
cock fritters and crab cakes and so stuff like that. So I miss being home.
In a long time. Some more of those little burrows coming out.
Is there something in it?
Cracking.
That's a big burrow.
It's close to bed. We watched the West Palm Beach. It says close to Ben's house.
West Palm. Unknown explorer. Texas. Drop bears.
They're nasty.
Just show me. Just like that. Oh, okay. That could be fun up there, huh?
Drop bears do not make. Drop bears do not make murals. Noted.
Little shrimp. Big shrimp? Bit little.
Tasty.
Tasty. Just had one.
You haven't seen it in Australia. Yeah.
What are those things that you saw on that island?
Raucous.
Raucous.
Kangaroo rats.
Kangaroo rats.
The combination of kangaroo. Isn't there a combination of kangaroo and rat?
Yeah. Yeah, cool.
Yeah, they're pretty cool.
What was the name of that island? You know what?
Rottnest Island.
Yeah.
What's that?
I miss Western Australia. That was fun.
Good place.
Yeah. I mean, I like Eastern Australia, but I think we just have more time to hang out with them
Yeah.
before they will come.
I love Fremantle.
Go.
There's a lot
like
not to see up the coast.
I see going that direction. That direction. That direction.
That's fine. I'll slide back away. I just want to see what this mass pulls into. Still counts as transect.
Oh, there's a burrow.
What's that?
Several of them. Oh, there's something.
We found
Is that a lobster in there? Something.
looks like a bunch of shrimp. Oh no, it's just one of those big lobsters.
Yeah.
Well, what's that?
Is he living there or is he food?
Either made it or took it over.
Mm.
Wow.
We can go back almost no transit. So we go back to that.
Keep going. We're about what, 25 to go?
Not even.
So, if you're just joining us, this is ROV Dive is part of the Northern Transects, depths of the Northern Great Barrier Reef. We're currently doing a transect at Noggin Pass in the SuBastian ROV. This Noggin Canyon is part of the Central Great Barrier Reef, and at present we're just coming to the end of a 500-meter transect for collaborators at Geoscience Australia for their benthic habitat mapping. So another 10 meters or so. We're about 680 meters depth, 7.7 degrees water temperature.
That's about it.
that a sea star? Crinoid.
And transect.
And we are done with the transect.
That's cool.
So there's a crinoid?
Yeah. So
Facehugger.
No, sea star.
Sea star? Brittle star? No.
See that
There's a
mark?
worm
Yes,
retracting.
That's where the longworms suck you.
yeah.
Behind stuff.
So we're going to just keep moving up the canyon for the time being. See what we can see. Quite a vast soft sediment substrate here. Probably mostly fine sediments, silts and clays, maybe some sand mixed in amongst it. All sourced from the Great Barrier Reef, which is just above us. As it's transported off the continental shelf through the gaps between the coral reefs, which is what's at the top of this particular canyon, Noggin Canyon. So now that the transect's over, we've got more freedom to look around and what's that? It's an isopod, giant isopod. So this is a large benthic arthropod, an isopod. This is some of those larger burrows that we were seeing with the sort of dome-shaped opening and a flat base. These are most likely the culprit that's making those particular burrowings in the substrate.
Is he eating mosquitoes? Just want to go.
How big is it? Those lasers are 10 centimeters apart, so that gives us a scale. So he's about 20 centimeters, which for a large cockroach, essentially, that's about the size of your forearm. And it does look like a trilobite, although trilobites, they're arthropods, but they're an extinct group of arthropods. They were dominant in the Cambrian through to the Permian, went extinct at the end of the Permo-Triassic extinction, but probably performed a fairly similar role to these isopods. Very good scavengers.
Just prettier.
Vicky says trilobites were prettier. They had better eyes, no doubt. Better on the eye, perhaps. Yeah, I think he's alive.
Maybe carry around a stick.
It could be a molt. So all arthropods molt their skin. They have an exoskeleton. They have jointed bodies and segmented legs. So that's common amongst all arthropods. So you can see the different segments of this particular guy. You can see the joints in his legs. In this case, 6 or 10 legs, I'm not sure. He seems quite happy, not moving around, so.
There you go. Shoot. Go a little bit north to left.
So whilst this might look like a bit of a desolate landscape, there's actually quite a lot of things that are going on. There's lots of bioturbation. You can see all the disruptions in the surface of the sediment. Each one of those is done by some organism. You can see trails, so squiggly trails left in the sediment. We see the odd. We're probably scanning a little bit high to see a lot of things, but there's probably quite a bit of smaller flora towards the base. Starting to see a few xenophyophores pop back in, so those large benthic foraminiferas, agglutinated benthic foraminiferas, sea star. So we're just on a wander around this depth. So we've seen quite a few of these large divots in the sediment, which appear to be made by mostly those isopod, but some modification from sediment and surface currents. So we're just going to settle down here for a second. We're going to take a couple of sediment push cores. Of around 25 centimeters of sediment, hopefully. So those large burrows that we're seeing of the isopods, they hold their shape quite well. So kind of expecting quite a stiff sediment. So something that should hold together pretty well in the tubes. This is a nice cohesive core there. We'll be able to get that back into the lab, use the sediment to do analysis on the microfauna, perhaps date the age of the sediment and see what sort of sedimentation rate we have in this particular location.
I'm gonna go 5.
Okay, that was 3.
Cody doesn't like these numbers.
Um,
Oh, it says 6 on there. You okay with that?
Sure it's not a 9?
you want that?
Yeah. Looks like this tube has a 6 right now, so it's 5. Very nice core.
It wasn't until you lost it.
No, that's fine. Yeah. It looks like we managed to sample some fauna on top. Not sure what it is. It looked kind of tubular, could be a bryozoan.
a zone.
Is that what they're saying?
Zone says by zone, yeah. Brian's not here. I'm sorry, I was turning the lights on so you can see. He knows.
Is that from us?
It's gonna be on screen at all times.
We're trying to get each other covered.
Lots of marine snow in the water. There's little particulates. Which could be sediment, could be organic, could be animals, could be plankton. It's a flat-headed fish.
Oh, it's official.
It's official. It's official.
That's a fish. It's official. It's official that you're better.
And coffin fish. Puffia. Could be. Seems there's a lot of fish experts on the YouTube today, so
Footed fish.
coffin fish. Thanks, Daniela. Oh, he's got a lure.
Trying to scratch his beard on the scene of 5 door.
Oh, he's waving.
Kind of looks like getting in the
Oh, yeah.
Oh yeah.
You go bike it? He has never seen anything this better before in all of his years.
Well, if he's a bin fish, he probably builds like
He's frowning.
Yeah, he's not approved.
Yeah.
Are you percent?
Yeah. Do your thing in the day.
Hook him up.
Is that a lure?
You can see him flapping it around?
Yeah.
The red-footed walking
Angler, frogfish. Monkfish?
meatfish, redfish, bluefish.
One fish, two fish.
Who's hunting?
I think he did. That's all he's got.
That's all I got. That's it.
Tired.
Staring competition.
But he really wishes he had pilots right over there. show us here.
Whoa, what is that? That's unnatural, that's unreal.
There's sweets showing the tail, big foods.
Oh, what have we got? Is that another one of those sea cucumbers that's really transparent?
That fried rice is on course.
I like that fried rice.
Yeah.
Yeah.
Kind of tastes like jambalaya.
I liked the teriyaki tofu.
I put the teriyaki sauce on the fried rice. They don't go for the soup? The vegan
No.
one, yeah.
Oh,
it
no,
broke my
didn't see the
finger, but I
soup.
grabbed this ladle. Like,
Yeah, me too.
Power too, but I didn't have it. That was so good.
The salad was even good.
Yeah. Got a little bit of garlic right now. Not as much as I usually do on those.
Another fine lunch on RV Falkor.
Where it like burns. It's like wasabi. So much garlic.
Oh,
This is a sea star. Sea star like this.
yes, it is.
Yeah, do you know sea stars start off just like
Yes.
Start off just like we did.
Get that fly through.
well, we formed
It was up before.
spinal tube, neural tube. Is that what it's called? Neural tube.
You're the neurologist.
And we form around it. And
Yeah.
go from there. And that's what spina bifida is. So it doesn't fuse the involving vegetation. Please don't. Try to act smart.
Yeah, you went to school to be that. What'd you go to school for?
Brain stuff.
Stuff's in the brain.
That was over 10 years ago.
10 years ago. I was 18.
Giving away all your secrets now, Cody. Is that another isopod? Or is it one of those? No, I think it's one of those holothurians. They're very transparent.
Yeah, that's yesterday. Oh,
Should we see if anybody's at home in that burrow?
Okay.
Oh, there's
Oh, yeah.
another one. That one's empty. Is there something in there?
It's like a fresh excavation.
Hmm. Who's at home?
They're
Oh,
fishing.
got a little fish in there. So, these look like isopod burrows, but clearly the isopod is not home today.
got it.
He's either evicted or he's got a roommate.
Pretty shy roommate.
Yeah. Oh, he doesn't want to say hello. Looks like the bioturbation is changing a bit. Getting these little teepee mounds. Bioturbation. Bioturbation is where the sediment is getting mixed up or churned up by critters living in the sand or in the mud. So it can be burrows or it can be these little mounds
Or trails.
or trails. And they might be residential burrows where something is living inside it, or they might be feeding burrows where something's just been in there feeding.
wants us in closer.
Jeremy wants corals.
Jeremy would like a close-up.
Jeremy's ready for his close-up.
Is that close enough, Jeremy? So we're zooming in to take a bit of a closer look at this sediment substrate. Oh, speaking of zooming in, Jeremy. So one of our colleagues onshore, Jeremy Horowitz at James Cook University at Townsville, is interested in some of these little cryptic. Well, he's specifically on the lookout for well, black corals is his specialty, but also tiny little sponges that we're collecting if we see some for our colleague Merrick Atkins at the Queensland Museum. And some of these things are quite tiny, so we need to get quite close to the seafloor and zoom in.
Apparently he's quite busy today.
Probably.
That's one of the Echeirid worms. It looks like it might be the discarded tube. So one of the challenges for the scientists that are on board the ship is that we are tasked with collecting samples for our colleagues on shore, and we have no expertise whatsoever in the types of biology that our collaborators and colleagues are wanting to collect. So we're being guided by messages on our chat channel that we're using between ourselves, and furiously trying to Google what things look like so that we can try and keep our eye out for them. There's something sticking up there. Good view down a burrow.
Stronger. Very much.
What is that? A shell? Discarded shell? Mollusk shell? An empty shell. I don't know what type of shell. Oh, it's got a.
It's been eaten.
Looks like it's had a chunk taken out of the side of it.
Yeah.
Yeah.
New
Might have been. It might have been preyed upon by something else. Carrier shell, Xenophora. What's that?
Slide to the right. Slide to the right.
What did you spot?
The sonar is also known for
Okay.
they work. I got closer to
Not a thing.
Sometimes some of the returns in sonar look a lot better from a distance.
What's that? What is that? What did you say? Oh yeah, is it shrimp? Okay, check him out. Oh, what's that?
That one.
Oh, that? Yep. Let's go check that out. Pearl? Uh-huh, coral. We're on the coral reef.
Another flabellum.
Flabellum? Two is always better than one.
It is.
May we collect this flabellum, please?
You can have whatever you like.
And, really?
Is it moving? Or is it just
Waving?
flapping in the wind?
Mm. So we collected a flabellum earlier, and we're going to collect one more in case we accidentally destroy the one that we collected the first
Something
time.
always goes wrong. Yeah, hey, Brett says collect it. So.
Hi Brett. Yes, we'll collect another flabellum in case you break the first one.
Take a look around that.
A pretty picture of that. Very nice.
So these are azooxanthellate corals. So they have no symbiote in them, no symbiotic algae like the shallow corals do. It's still a scleractinia, which are the hard stony corals. So he's got tentacles, he's got a mouth, he's got a stomach. It's kind of like a jellyfish with a limestone cup, and the limestone is the skeleton. So he's retracting his tentacles at the moment because he feels threatened, as he should. So these corals are related to jellyfish. You didn't know that? They're all part of the same phylum. They're all Cnidaria, it's spelt with a C. And what they all have in common with anemones and hydroids and jellyfish is stinging cells called nematocysts. Ooh, there we go.
It's a good size.
Yeah.
Thank you very much. Give Sebastian a little shake off.
Box B. Yeah, got that. I was distracted. I was watching the coral.
Might have to do with those sonar.
Is he flexing? Is that what he's
It's called
doing?
reverse cobra.
Showing his guns.
I want to take a selfie.
Ah,
Just got no idea.
go. Is that another one?
Yep.
We could,
And if
we
you
could
ski,
probably get one for you, Ski.
I don't know if there doesn't seem to be a lot of other options. at the
No,
moment.
that's all right.
They should do it after both.
Sorry? Oh, okay. On the everyone that's watching, we're just wondering how the audio is on your live feed at the moment from here. If someone in the chat can give us a comment, or on the Facebook or the YouTube. Are we coming through clearly for you at the moment? Audio is okay? Yep. Sounding fine on YouTube, all good? Excellent, good to know, thank you. Loud and clear. Oh, there we go. So we're just going to zoom in to see if this is another one of these flabellum cup corals. It is. So we've already collected 2 on this dive, and those are for ourselves, and we want to preserve them in a particular way and fix the coral tissue and intact with its skeleton. But we have another collaborator at the University of Tokyo who is also interested in these types of corals, and he's interested in the skeleton material, and he would want them preserved, or not preserved at all actually. He'd want them treated in a different way, so he won't be able to use parts of the samples that we've already collected, and so we will take one more of these while we have the opportunity. Bless you. If you were tuned in earlier and Luke was describing these flabellum corals, he was saying that he's been trying for a decade to collect one of these. So now we have 2 for us, and we'll get one more for our colleague Yusuke Yokoyama at the University of Tokyo. And he's interested in doing geochemical analysis on the calcium carbonate, the limestone skeleton. He'll be wanting to date the age of this. And other trace element and isotopic geochemistry. So the way that we're going to preserve the samples that are for us will mean that he won't be able to use that. I would have to go in a separate spot, so it'll have to go in 1C, please. And then we'll leave flabellum alone after this. We won't collect any more. There we go. Thank you very much. Should try and have a look around. I know Jeremy's looking for little things that will only be really tiny and we'll only see them if we're looking in the sediment. Yeah. Just have a
That's zoomed in or
close.
more zoomed in?
This is good to look around. What is that? Is that like the thing
That's
that
the
we
thing we
collected?
kept before.
Can we have a close look at that little thing that was waving around? It looks like what was
I think
on
that's-
top of the core that we collected earlier. Interesting. It does look a bit bryozoan-ish, doesn't it? But it's so covered in sediment, like smothered in sediment, which for a filter feeder is not optimal.
I wouldn't think so.
Yeah, yeah. But then look at all these tubes. It's weird.
No, I don't know.
And it has a little shrimp friend. What are these little things sticking up? What are
Big worm
these?
heads. There's a lot of them.
There's a lot of them.
There's a fish.
Ah. Curious. So is that as close as we can zoom?
Not close.
Close as we can zoom? Okay.
I guess I can get all the
Nudge forward a little bit. And we're curious about these little knobby sticky uppy things too. We have no idea what they are. Oh, hello.
Whatever.
anything.
And then we should and then after that, if we could take a look at that rim there, at this, whatever's sticking up there.
Talking about out there.
Yeah. Yeah. Thank you. Yeah, we're curious as to what this thing is. I missed that.
Merrick.
Oh, Mary. Just let the dust settle there. It's interesting. Looks like it's, you know, it's fan shaped, which initially made us think of a bryozoan, but it looks like it's constructed of tubes. And bryozoan tend to have a low for 4, so they're a filter feeder. So I would have thought that this muddy coating would have been suboptimal for something that probably doesn't want to get clogged up with sediment. Just trying to have a look at the various chat things that I've got open to see if anyone's giving any suggestions.
Scaffold behind it.
Almost looks like polychaete tubes.
Oh yeah, a collection of polychaete tubes. Is that what you're suggesting, Vicky? Yeah,
It's agglutinating.
yeah, like a,
Polychaete tube.
yeah, it's interesting.
And down here you can see them as well.
Mm-hmm. Well, it's not something that we're going to collect.
Want to go look at that in the room?
Yeah. So what we're looking for in this close to the seabed are these little cryptic things that we're zooming in on or trying to find are tiny little carnivorous sponges. And we will take the opportunity to have a look at the fish. And there's an anemone. Hello to Merrick Ekins from the Queensland Museum, who's watching online.
Hi, Merrick.
And Merrick is our sponge specialist.
No.
And he's very interested in these small carnivorous sponges, which seem to have quite diverse morphologies. So I'm not entirely sure what we're looking for. And hopefully Merrick is watching in real time and can quickly send me a chat message saying, yes, collect that, if we see the right thing. He says it looks like a dead octocoral. Let's just keep panning around. Yeah, we also don't know what these tiny little black nubby things that are sticking up out of the sediment are either. Nice fishing in. Let's chat for a bit. I'm just going to message Eric.
Fresh one sucker. Lemonade. Sure.
This one.
Dear, you want break?
John, please post the Zoom meeting ID back in the ROV channel.
Am I breaking this?
Merrick might join us.
Is there other things we move around a bit?
We can continue having a look around while we see if Merrick's going to join us.
So are these the sorts of things we're looking for?
No idea. Should we go and say hello to this worm while we're seeing whether Merrick's going to join us?
Yeah, just have a look around those.
Oh, you want to look at what those
Well,
things are?
around the worm end.
Yeah.
Update.
This is one of the echeurid polychaete worms that we were talking about earlier. We've seen lots of these on this dive. Oh, okay.
He replied to your comment instead of in the chat,
I see.
which I didn't know was a thing until I missed like 23 messages or something.
It's a lot to keep track of. A lot of different chat feeds going on. You gonna poke this guy? Give him a poke? What? No? No? What? Oh.
Yeah. I want to find some sponges.
We'll give this a little caress.
No.
No. Okay. So this must be a discarded sheath or something like that. Oh, hang
Oh,
on.
no.
Wait. Oh,
There we
no.
go.
There we go. There we go.
It just takes a towel.
These little black heads are everywhere.
Yeah.
It's
Oh,
okay.
no, yeah, no, yep. Oh, there we go. Fist bump.
Did one just float away?
All right. We might leave him be.
No.
Looks like he's had enough. Yes. everyone that's watching online. It is all good here. We're just having some, not issues, we've just got a collaborator on shore that's trying to zoom in and join us on the commentary, and we were just in the process of getting that set up, which is a bit of toing and froing and audio check and audio testing and what have you. So we didn't want to bore you guys with all of that, so sorry it was a bit quiet there for a little bit. Okay, and he's just about to ready to join us now. Hello, Merrick. Are you? Okay, all right, we've got that sorted out. So we're being joined live or remotely via satellite by Dr. Merrick Ekins from the Queensland Museum, who I believe is the curator of sessile invertebrates. Is that right, Merrick?
Yeah, question manager of sessile marine invertebrates, so all things that are stuck in the bottom of the ocean and don't move around too much, so yeah. And on this particular expedition and other ones, I'm particularly interested in carnivorous sponges. So most of these guys are really tiny, probably most of them are only a couple of centimetres, if that. Some of them are even 5 mils long, so they're hard to spot. And sponges normally are filter feeders, and these guys have decided to take this unique opportunity because there's not a lot of food down the bottom of the ocean. So they've got these, most sponges have silicon spicules, which they use for structure and support. And some of them are really super tiny. They're probably like 5 microns in size. And they have these ones that are shaped like little hooks. And so these guys have done an exaptation where they've decided to use these to capture food. So little tiny crustaceans, isopods and things like that get caught. Their fine hairs get caught in these spicules, and the sponge decides to digest them rather than filter feed lifestyle.
And is this the sort of habitat or the sort of substrate that you'd be expecting to see them, Merrick? We're in a very soft sediment substrate here.
There are a couple that are in soft substrate, but most of them tend to be attached to rocks or even some other sponges and things like that, hard things. And they produce a bit of a stem to get away from all the raining silt, and so then they can be out in the water column. But there are some like the beautiful lyre sponge that's been found in sediment, and I did find a few other ones on another expedition off the east coast of Australia down at 2,000, 4,000 metres. So
that is quite a bit deeper than what we're at the moment. We're at 683 metres, and it's 7.3 degrees Celsius temperature here at the moment.
That's quite warm then.
Yeah, quite warm. So we collected some samples for you on the previous expedition. Would you like to tell us a bit about what we found, or what you found from the samples that we collected, and what they were, and if they were something new for you?
Yeah. Well, it looked fantastic. Yeah, it looked like we've got at least 3 new species of carnivorous sponges there. Unfortunately, we just at the Queensland Museum here, we just had all the offices and labs renovated during COVID. So we're just allowed in this week, really. So I haven't had a chance to fire up the scanning electron microscope, which is what I have to do to look at the fine detail of these spicules to be able to work out what they are. So I'm champing a bit to get there. But yeah, and, some of them look fantastic. Great job, guys, who are on the boat. Thank you very much.
Yeah, oh no, it's exciting for us too, especially, well, those of us who this is not our field of research, these are not our specialty, but it's really interesting and really exciting for us to be a part of these sorts of collections as well, even though it's not ourselves that are going to be doing the science. We're all curiosity driven, and the chance to collect something new is really exciting for all of us. So it's a pleasure to be on the boat and have the opportunity to maximise the ship time and collect samples and data for as many collaborators as we can. Yeah, so Merrick, you can go ahead if you want to direct the ROV pilots in any particular way, if you want us to slow down or speed up or zoom in, feel free to give a bit of a direction. We're just doing free exploring at the moment. We've finished our transect. So the business end of the dive is kind of finished for us, and now we've got a little bit of time to just look around.
Yeah, it's good to have a look around. I was going to say if you can see any sort of rocks or structure, that would be where I'd say go ahead there, but I guess it's all on a sandy ridge at the moment, isn't it?
Yeah, on this dive we really haven't seen any hard substrates. There weren't any rock outcrops sort of at the bottom of the dive towards the base of the canyon axis. It was all has this soft sediment drape over it. So really we've been in this sort of bioturbated sediment pretty much for the whole dive. There really hasn't been any hard substrate at all, which of course that changes what types of biota that we're going to find here.
Yeah, that's right. I mean, the ones that do occur in this soft sediment, there's like one called the lyre, which is quite wide. I think it's about a foot wide. You'll see it, and it's trying to support itself on the structure. It looks like a big X usually with all candelabra sort of structures sticking up. But some of the other ones, like there's little ones that are not carnivorous, but little things called radiella, which look like upside down cones, and they're in the sediment, and they're only a centimetre wide, so they'd be incredibly hard to see unless you stop them. Probably scoop sand is more likely to find them.
I mean, we can certainly slow right down and pan around and zoom in as much as we can on the sand, and we can just sort of pan around and have a look around and see what we see. So do shout out if anything catches your eye.
Don't worry, I will. Yep.
So I think the concept of a carnivorous sponge is probably something that's new to most people. Can you tell us a bit about them and a bit more about them and how they feed and that sort of thing?
Yeah, well, they were only discovered in the mid-90s and from a cave in the Mediterranean. And I think that's why people had found them 100 years ago from the Challenger expedition in the 1890s, but they didn't realise they were carnivorous sponges. And it wasn't until someone doing some scuba diving in these caves in Europe and actually went, hang on, it's actually catching things, because you pull up your sponge and it would have these crustaceans on it, and you think, oh, well, they just died or just got caught in the trawl or whatever. And it wasn't until someone looked at it in the 90s and went, hang on, they're actually carnivorous. And so since then we've gone back and looked and worked out, okay, with a bit of molecular help and classical sponge taxonomy, looking at these spicules, you work out, okay, this whole family, we think this whole family, Collaborators, is actually all carnivorous sponges. So, in a way, it's a bit more sexy, a bit easier to sell than a normal sponge.
It certainly sounds really impressive.
So, getting back to the question about biology, it is not quite in their charismatic megafauna grounds, but closer than trying to sell a sponge to someone. I mean, sponges, we don't know so much about sponges, like just from around Australia and predominantly Great Barrier Reef and normal scuba diving depths. In the Queensland Museum here, we've got 5,000 morphospecies, but only 800 of them have been described. The rest are all potentially new to science. But because they're not super sexy, it's a bit hard to sell to the politicians or public. Oh, can you get some money to do some taxonomy on sponges? But with carnivorous sponges, maybe there's a chance.
Yeah.
But in terms of biology, normal sponges are just big filter feeders. They just take the ocean in and filter it through a little flagella and out the other side and filter out anything that's food particles. But these ones down at the bottom of the ocean, and even down here at 600 metres, there's not as much food as there is in the water column at, say, 30 metres on a normal reef. So that's why some of them have opted out to take and eat basically whatever comes past. And they're not as dramatic as like a Venus flytrap. They don't suddenly go chomp and grab something that walks past. Basically, if an isopod or amphipod is walking over the sponge, and they all have a whole lot of basically like hair-like tendrils hanging off them, And these get caught one at a time until enough of the hairs get caught that the animal gets trapped and can't move. And one really cool thing I love about sponges is they're totipotent, which means it's a bit like stem cells. Each cell can go off and become something else. And the classic sponge show and tell thing from, I don't know, 100 years ago, they used to get a sponge and you'd mash it up in like a blender and then strain it through cheesecloth. And if you put it all, provided you kept it all in like a nice saltwater solution, the sponge cells would all get back together and start to organize themselves into organelles, into a sponge sort of shape. And that's what these totipotent cells are. They can, like stem cells, go, okay, you'll do this and you'll do that. And that's what happens when one of these crustaceans caught. The sponge will basically go, okay, you cell over there, you're going to now be a digestive cell. You're going to go out there and you're going to digest these amphipods or whatever. So that's how they work. It's pretty funky. I think I would prefer it if it did something like a Venus flytrap and went and jumped. That would be even better. But maybe there's one out there that does that. We just haven't found it yet.
And have you always worked on sponges, Merrick, or have you worked on a bunch of other stuff as well? And we're just zooming in to have a look at this flabellum here, which has been of interest to us this morning too, but we've already collected a couple, so we're just going to have a look at this one, but we won't be picking it up. But yeah, what other sorts of groups do you work on or have you worked on?
Well, in a former life, I was a plant pathologist. So I used to work on fungi and sunflowers. And then I decided, you know what, I think I'd rather go scuba diving for a job. So I changed to marine biology. And then, yeah, as well as working on sponges, I also work on ascidians, sea squirts, and octocorals, another group I work on. But I'm also in charge of other groups like bryozoans and hemichordatas. And we do a bit of work on the hard corals as well. But there's plenty of people at MTQ working on the hard corals, so I can work on the soft corals, octocorals. But carnivorous sponges are my favourite at the moment.
They certainly sound very cool. Merrick, do you have any idea what these little black nubby things that are sticking up out of the sediment are? We've been seeing them for quite a while, quite spaced out. Evenly spaced.
That one there you're talking about on the screen there?
Yeah, they just look like little black dots, really. It looks like it's the tip of something sticking out above the sediment water interface. Do you have any idea what those are?
Yes, it was a worm tube.
Yeah, that's certainly the tip of a worm tube that we were just on then.
That's what's great about having the ROV. You can actually go and look at something and you can actually collect it. Within limits, of course.
Yeah, that's right. It's a bit different to taking a dredge sample or something like that, isn't it? Where you bring up a whole bunch of stuff, but you really have no idea of the context of where it was situated or what it was hanging out with or what was the substrate or any of that sort of thing. So
yeah, and what it even looked like. That's what's great now. We can get images of what they look like. Because a lot of things, especially down a bit deeper, they come up from 1 degree. So even by the time they get to 20 degrees, they're basically parboiled. So a lot of things will change colour as they're cooked. Yeah. And sponges in particular, a lot of them will just collapse in shape. Like, for instance, there used to be one of the first, what we now know as carnivorous sponges, was collected, and it had all these, when they were brought up from dredges and that, they just had these flattened sacs, and no one quite knew what they were for. And it wasn't until recently that they got spotted with an underwater camera on ROV, and actually they're in these inflated spheres which the sponge used to hold itself up above the sediment. So that's really cool. Now, after that expedition, the ones you collected before, and they say, yes, I've got images of these things for the first time. As you were saying before, when they come off a dredge or a trawl, it's like they've been in a washing machine with everything else that's come up, every other crab or sponge or coral or whatever, until they all get a bit mashed and damaged. So it's really nice to see them intact and then collect them beautifully. The pilots are doing a fantastic job collecting these things.
Yeah, absolutely. I know that tiny little carnivorous sponge that we collected on the last expedition for you that looked like, well, when we were collecting it, we had no idea what it was. It was on that stalk looking like a little daisy or sunflower or something like that, and I think we collected a few, but we managed to destroy a few in the lab when we were trying to preserve them as well. So fortunately we have the 4K video that you can go back and look over and see exactly how they were in situ at the time when we collected them. But yeah, having the ROV just is a whole other dimension, isn't it?
Yeah. No, it's fantastic imagery, and the skill with the guys can collect it. It's excellent.
Some very skillful flying going on right now at the moment. We've had a change in the pilot seat, and we've got John, our lead marine technician, is having a fly of ROV SuBastian, which I didn't even realise at the time when I was chatting away with you, Merrick. It was all going so smoothly and seamless transition. I turned to my right to say something to Cody, and it wasn't Cody, it was John. So normally John sits in the technician chair at the end of the bench here, and he's doing all the audio and video, and got many screens open with lots of buttons to push and what have you to keep all of this looking seamless and effortless. But I can assure you it's not effortless at all. There's a lot of running around and tech and what have you that goes on in the control room behind the scenes to bring all of this live stream out on YouTube and Facebook for everybody to be able to join us and get involved in the science and see it all unfolding in real time as we do. And the ROV pilots and the technicians just make it look they make it look easy and seamless, but I can assure you there's a great deal of work and running around involved and skill and experience. So that's wonderful for us. It's great, makes it very easy for the scientists to just sit here and do our thing.
They do a great job.
So we had a little earlier start on our dive this morning than what we were anticipating, so that means that we've got a little bit of extra time at the end here before we'll need to end the dive and bring SuBastian back up on deck. So we've taken the opportunity to get John out of the technician chair and put him in the pilot chair. Any of our fish friends online able to tell us what the sky is? It's like he's watching the paint dry or something. Silly fish, yeah, he looks a bit silly.
It's great. Looking straight down and looking for something to feed on.
He's looking for carnivorous sponges.
Finds one for me, great. I'll keep that fish alive.
Merrick, sometimes we have some interesting conversations between ourselves, scientists, about what's deep and what's shallow. It was quite interesting on yesterday's dive. We were sort of in the lower to upper mesophotic zone, and Tom Bridge was joining us, and for us we were getting blown around in SuBastian with in the currents, and we were talking about it was problematic because we were so shallow, and then Tom started joining us for commentary talking about being in the deep reef. So
Yeah, that's right.
it's all relative. So what's deep and what's shallow for you? For your ideal sponge targets, what's deep and what's shallow for you?
Yeah, that's right. Probably 2,000 metres to 4,000 metres is ideal.
Yeah, that's deep.
The scuba diving depth, which is normally up to 30 metres, is also another favourite. Yeah.
So I guess on today's dive we're somewhere in between those two extremes.
Yeah, that's right. Yeah, well, nice to get hold of some of those rigs that other people use and go down to a couple hundred metres. Yeah. Oh, nice.
Look, I think that's a cutlass fish. Merrick, I have a question for you. So, I mean, you work at the Queensland Museum, and for most people, their experience of a museum is going to visit the museum and wander around and look at the exhibits and what have you. What's life like at the museum for you? I imagine that you have an office and a lab somewhere in the back rooms, or in the dungeons, or the deep dark depths below the museum. How does all of that work for the curators, and what's your job, and what's a typical kind of day at the museum for you?
Yeah, well, that's right. If I think of the Queensland Museum at South Bank in Brisbane here, you know, it's got 5 stories and it's got 3 levels, all public galleries, and then it's got another 2 levels above that, and one is cultures and histories and administration, and on the top level here is biodiversity. And so we've got a whole bunch of offices and labs, and we've also at the back, we've got what you're just calling the dungeon, the spirit store, where we keep several million specimens in ethanol, such as like the carnivorous sponges we collected before, and that's what we call the state collection. And so we're trying to get all that stuff online as well, so people can see it. So the normal public can't see it. We can get other scientists, and you can make appointments to come and look at things as well at the back. So it's a fascinating time. A normal day for me, well, usually I have these great plans of what I'm going to do every day. Then I turn up to work and then I check my email and off that goes on another tangent. So a lot of what we do is public inquiries. Usually with sponges, I don't have too many public inquiries, whereas the guy sits next to me, Jeff Johnson, the ecologist, he gets lots of public inquiries about fish. Whereas I get a lot of scientific inquiries from people all around the world trying to identify sponges usually. A lot of time, at the moment, I'm identifying some sponges and ascidians for a few PhD students around Australia who are working on other projects, and they pull up some sponges and go, oh, what's this? And with sponges and ascidians, there's no reference book, like you can't look up a bird book or a fish book and go, oh, that's what that is. They just don't exist yet. No, only recently has there been a 2-volume massive book, 2 books on that just cover the main genera of sponges. And since then we've done that, as like I said earlier, we've got 5,000 morpho species in the collection and less than 1,000 of them have been described. So there's so much that's unknown. So I spent a lot of time doing that. And unfortunately, to identify a sponge, bar probably about half a dozen, maybe like glass sponges, we might see some of them later, that have fixed state. The rest of them sponges have a sort of amorphous sort of shape, so it could be anything, and you might really go green sponge, red sponge, blue sponge. But to identify them, you have to look under a microscope, and to do that, you have to make microscope sections. So that's usually a 2-day process just to make the slides, and then looking at them takes another few hours for trying to identify something. So it's a big job. So people love to go out and go, I've collected 200 sponges. Can you tell me what they are? And I go, well, that's a couple of years' work there, sorry. So, but I help out people wherever I can to do that. So that's a lot of my time. And I've also got a lot of time to spend, a lot of time that I do spend registering the backlog of stuff. Like there was stuff that was collected over the last 100 years. So Queensland Museum is young compared to other museums like the Paris Museum and the British Museum, which have, you know, they've got crates of stuff from Charles Darwin and so on that haven't been open since then. You know, no one's had time to go around and look at them. And that's what happens. So we spend a lot of time working on the new stuff, and we try and work our way back through the old stuff as well. And a lot of the stuff in the past people might have collected and they might have gone, oh, it's a coral and it comes from Australia, or if you're lucky, it might come from Queensland. And so, you know, it makes it harder to identify, but it also makes it less valuable in a way, because what we really like is really good location data. And these days, of course, I love photos, because a lot of that stuff in the past, collected 100 years ago, there was no photographs of it. And if it was brought up like a dredge, you know, it's lost its colour, it's lost its shape. So now we're trying to get material that we can photograph and have images of, so we can say to people, this is what it looks like now. And also these days, we want stuff for DNA. So a lot of that stuff that was preserved a long time ago was, and hundreds of years ago, all they would do was dry it. But for the last, within the last 100 years, they would put stuff in formalin, which gives you the shape and the structure. But it also means we can no longer do DNA stuff. And for a lot of these tricky things that don't have obvious shapes, you know, we do need the DNA molecular stuff to help us along the way. And that's another thing people say, well, why don't you just sequence it? Well, in order for a DNA sequence to work, you have to have a skeleton to attach things to. You have to know what the original is there. And a lot of these species that were described hundreds of years ago, we can't get the DNA out of it because they're old and dried or preserved in formalin. So we have to try and find a modern equivalent, you know, try and find that and prove it's the same as the original description, and then we can get the DNA from that. And then we can start making this tree that then later on we'll be able to attach things to. So that's a very slow process, but we're working away at that at the same time. So that's another thing we do, molecular stuff as well. So,
yeah, I can see why it's a career's worth of work to try and work through all of that new stuff and old stuff.
Oh, it is. Oh, well, yeah, we always joke that we've got 10 lifetimes worth of stuff just to deal with already. So, and it's really good in an ROV expedition like this one on the Falkor using SuBastian, is that you just collect a limited number. But if you can imagine someone does a trawl, like a lot of the time in the past, and it still goes on, like CSIRO and Department of Primary Industries will do like a bycatch survey. They'll go, okay, let's hire a trawl and tell us to just do it one trawl and bring everything that it gets on board. And you go, okay, well, within that 1 hour, you've collected potentially thousands of specimens. So that's potentially several years' work already just to do that. So I love trips like this where you just targeted a few things, and it makes it easier to finish off and work on the next project. But
yeah, exactly.
New species, that's my favourite bit.
Yeah, and it's great to have that continuity. It's great to have that continuity between Jeremy and yourself from the previous expedition, and we can say, well, we've already collected one of those, or we collected 2 of those on the previous voyage, so we don't need to collect another one of those on this trip. Or, you know, on the previous voyage we got A, B and C, but we're missing D, so we're just, on this trip, we're just going to go looking for D. We can be quite selective in what we collect.
Yeah, that's right. Yeah. The chances are the carnivorous sponges, it seems to be, from my experience, each one we get is a new species, so don't hold back on the carnivorous sponges.
Yeah, that's amazing, isn't it?
Definitely is.
no, we'll certainly be
And
doing
because
more
they're
of
so
these.
small too.
Yeah, we'll certainly be doing some more deeper dives and some canyon dives on leg 2 of the expedition once we head up off Cape York Peninsula and the canyons and deeper shelf edge seascapes up there. We certainly won't get to 4,000 metres. The water's not that deep out there, but hopefully we'll definitely come across some of these deeper sites that have got a rocky substrate, and as you say, that tends to be where we saw the glass sponges and carnivorous sponges on the previous trip.
Well, that'll be great. I'll try and join in from several thousand kilometres south.
Yeah, it's really wonderful for us on the ship to be able to have extra people joining in and zooming and helping with the commentary. It's certainly, I mean, it's a pleasure and we love doing it, but it's a challenge to talk on the live stream for 6 hours or so, especially if it's on some of the biota that we're not so familiar with. So it's really great to have extra people like yourselves who are watching from on shore that can zoom in and chat with us.
Oh, that looks like a zoanthid. That's pretty cool. Yeah.
Yeah. Looks, is it moving itself or has it been dragged there? It's a worm, did you say, Vicky? A worm?
I don't know, it's probably on something else, possibly on the crowd. That might be one worth collecting if you feel like collecting, if you feel like still grabbing stuff. I have a Japanese researcher, Hiroki Kise, that we've been collaborating with and on the investigator trip in 2017, and we pulled up 2 new species of zoanthids from down there. And I talked before about going through the collection, and while I was cataloguing some old stuff, I went, oh, here's stuff that was collected in the Craigman survey where they did some deep water trawls in the 1970s. And there it was, but no one had looked at it, and it wasn't ever registered or digitised, so no one even knew it was there.
So, Merrick, is this something that you think that we should collect for your colleague on your behalf?
Depends on how much time you've got and how many jars you've got left.
Yeah, we've got plenty of room. We can pick that up if that's something that's valuable for you, for sure. And Merrick, if we manage to collect this for you, you'll just need to instruct us what we're doing with it in the lab. Is that something that's going into ethanol, or we can subsample some for ethanol and some to formalin? We can do whatever you tell us to do with it.
I think, yeah, they're pretty tough, so you can put them straight in ethanol. Yeah.
Almost looks like the colony is on the back of a. Well, we thought at first it was a worm, but maybe it's a snail.
Yeah, that's right. Normally a lot of these zoanthids are piggybacking on some sort of gastropod or something. So You get two organisms for the price of one at least. Love your work. Have you seen any stalk crinoids yet?
Not on this dive, no. We did see some yesterday, but we haven't seen any today on this dive, really. Mostly what we've seen today has been holothurians, shrimp, those Echiurian worms, quite a few fish around. Certainly at the start of the dive when we were at the bottom in the axis of the canyon, there were a lot of fish around. Yeah, not those xenophyre.
Have you been collecting the stalk crinoids?
No, we haven't collected any, really, on any of the dives that we've done. Not being, not really being any of our high priority targets for anyone else, but certainly our permitting allows us to collect Porifera, Anthozoa, a few other groups, but yeah, it's depending on what zones we're in. So yeah, we just need to make sure that we're collecting the right thing in the right zone. But if there's anything of particular interest, we can certainly have a look at the permitting and just see what we can do. So Merrick, we're probably going to continue this dive for maybe another half an hour or so. What would you like to do? Would you like to stay on the Zoom with us, which would be great, if you would. But just to let you know that we'll probably continue the dive for maybe half an hour or so, and then we'll be wrapping it up and starting to bring SuBastian back up and end the dive.
Yeah, I can hang in for another half an hour. I've got a meeting in 45 minutes, so that'll work for me.
Okay, yep, that would be great. So really we're just having an exploratory look around at the moment. We'll be keeping an eye on all the cameras on SuBastian. We can see behind us and around us and what have you, so we'll be checking out the water column, and we'll be looking at the seabed, the sediment as well, for those things that you're looking out for as well. But we've got a bit of free time at the moment, about for another half an hour or so before we end the dive.
Are we working our way up the ridge of the spine of the ridge at the moment?
We're working our way back to the top of the ridge line. Yeah, we're on a bit of a side of it at the moment.
As always on these expeditions, I was going, I wish I was on the boat, I wish I was there, seeing it all up close.
Yeah, I think there's a few people feeling like that at the moment. It was an amazing job by everyone involved to be able to pull this expedition together. Very short notice. We only had a few weeks to organise a team, a science team, and an expedition plan. Thanks to Dr Robin Beaman at James Cook University in Cairns. Has managed to put this plan together and make a few phone calls and a few emails and get a science team together. So, of course, because of COVID restrictions and border closures in Queensland and things like that, it was very restrictive as to being able to put together a group of scientists at such short notice that were able to come to sea and for quite an extended period of time. I mean, this expedition's been broken up into 3 legs, and in total we're going to be at sea for 47 days. So it's no easy feat to be able to organise things both at work and at home, to be able to come out for such a long time. So I know there are a lot of people who would have loved to have been able to join us on the ship, but it just hasn't been possible this time. And thankfully we're also able to connect remotely and still have people participating and being involved from onshore, even though they can't sail with us on board Falkor.
Yeah, I was going to come, but my partner had a previous trip away at the same time, and I had to look after our 20-month-old, so that was off the cards.
Yeah, right. Of course, ROV Falkor is a very nice research vessel to work off.
On the very first expedition where we weren't even allowed to collect at all, so that was very frustrating. So it's good.
Yes, I can imagine. Yeah, on that first expedition out to the Coral Sea, I was at home as well, watching those live streams on all of those dives. That was during the sort of earlier stages of COVID, when we were sort of in lockdown and weren't able to go to work, and everybody was working from home, and I was sitting at home. I'm finishing up my PhD thesis at the moment, so I was at home just writing. It really didn't impact me greatly as to whether or not I went into the university or whether I worked from home. So I set myself up in the living room and had the live stream going on the big screen TV. So I was sitting there all day, every day, tuned into those dives, and I can imagine whilst it was really amazing to have all the video and the imagery, I can imagine how frustrating it must have been to not be able to collect samples.
And how'd the write-up going? Are you still writing? Are you finished?
Still writing here on the ship.
Very good.
Yeah, very much at the pointy end. Yeah, I'm sort of tidying up my first full draft of the thesis at the moment to be able to submit that to my internal review panel. And very soon I'll have to give my final seminar. So I really need to get that draft completed in the next few days whilst I'm on the ship. Hopefully my supervisors and review panel will be very forgiving that it might not be quite the polished piece of work that I would have liked, but this was just such an incredible opportunity. There was no way I was going to say, no, I'm sorry, I can't come on Falkor because I have to sit at home and write my thesis.
You can sit on the Falkor and write your thesis there, yeah. Oh, that's great. Well, good luck with it. If you're almost putting together the final touches of the first draft, sound like you're most of the way through it. Yeah, good work.
Yeah, all the hard work has been done over the past 3.5 years, or getting close to 4 years now. Yeah, and certainly all those results chapters and what have you are done and dusted, and so now it's just putting it all together with a nice introduction and conclusion, and yeah, that'll be another milestone ticked off.
Can I ask what the title is?
Now let me think. The title is the Spatial and Temporal Evolution and Internal Sedimentary Architecture of Halimeda Bioherms in the Northern Great Barrier Reef.
Okay. All right.
So from looking at the Halimeda bioherms from a sedimentological perspective, and working on the geochronology of their initiation and development since the beginning of the Holocene, from the post-glacial marine transgression, putting all of that into context with shelf flooding and transitioning from a sort of a mangrove-dominated estuarine environment through to marine conditions really kicking off the start of the carbonate factory. The Halimeda bioherms, I guess, commenced quite a good while, about nearly 2,500 years before the nearby coral reefs initiated. So kind of putting all of that into a geological history and chronology. Yeah, we've been doing a bit of work on nitrogen isotopes from the Halimeda skeletal material. Sorry, Merrick. Whatever that was that we were just zoomed in on. Is that a target for Jeremy? Yeah. We'll zoom back in on that.
Yeah.
Now's not the day or time to be talking about Halimeda. That's a story for another day. Can we zoom in on it?
Like a little zoanthid.
Steadying here. Is it a zoanthid colony? Is that one of these zoanthid colonies?
Yeah, I think it's a zoanthid only.
So do they colonise over a substrate of something else that was there previously? So a stalk off of something else that's died and they've colonised over the top of it? Is that what that is?
They can do that as well, but they can also be like their own thing, if you like, their own organism. I'd say it might even be an octocoral as well. I can't quite count all the polyps.
Yeah, they're very translucent, makes it difficult to count. Yeah, we're thinking eight tentacles on each polyp, which would make it an octocoral.
Got very bad bandwidth at the moment, so I can't.
We've just got one of these swimming holothurians coming into view.
They were just such crazy looking animals, aren't they?
Oh, they're very weird. Sea cucumbers are weird anyway. So myself and Vicky Lowe, who's on Falkor from the University of Queensland, she's a paleoceanographer, we'll be on for the entire duration of the 3 legs of the voyage, 47 days. We've got Luke Notiff on here at the moment from QT. He's going to be getting off in Cairns tomorrow, so he's done this first leg from Brisbane to Cairns. And then we'll be coming back and joining us for leg 3 towards the end of the cruise. And then we've got 3 more scientists getting on in Cairns tomorrow, Rob Beaman and 2 other PhD students from James Cook University in Townsville. And they'll be joining us just for leg 2. So that'll take us from Cairns tomorrow up off Cape York Peninsula to the northern outer edges of the Great Barrier Reef and back down to Cairns. Then we'll have a Falkor crew change. Most of these crew have been on here for a couple of months now, some even longer, and they'll be changing out in Cairns and a new crew getting on, and then we'll head off and leave Cairns and make our way back to Brisbane for leg 3 of the expedition. The ROV team will be leaving us at the end of leg 2, so we'll certainly be utilising SuBastian and diving on leg 2. And then those guys will be getting off and heading home and having a well-deserved break. So on leg 3, when we make our way over the course of about 2.5 weeks from Cairns back to Brisbane, we'll be mapping and surveying the Tasmanted seamounts on our way back down to Brisbane, but we won't have any ROV dives on leg 3.
That's a shame. I need some video from the Tasmanted seamounts.
You should put in a proposal for a new voyage.
Yeah, that's right. I should. I've got a carnivorous sponge that someone collected off that I just described. Just got accepted a couple of days ago, actually. But it was collected from a rock dredge, of all things, over there. So they pulled it up on it. It was attached to a rock. And it was great.
Oh, well.
Another new species, but again, we don't know what it looks like underwater. You're on for the whole leg then? All 3 legs?
Yes, I'm on for the duration, as is Vicky. And on leg 3, we'll also be being joined by an artist at sea who will be joining us as well, which is really exciting. I think that adds a whole other dimension to a scientific expedition, to see the perspective and interpretation through the eyes of an artist. So that's going to be really exciting as well. I could just see a question popped up on the YouTube chat from the Unknown Explorer asking about Debs. How's she doing? She will be coming back on board Falkor after leg 2. So yes, she's doing well and has had a break and is coming back to join us again for leg 3. Oh, I'm informed that she's been remodelling her kitchen. So her holiday was not really a holiday by the sound of it. Oh, blowout. Rookie pilot.
It's good. He's clearing the top layer of sediment away so we can see what's under there properly.
You're doing a fantastic job there, John. You just keep going. Looks like we've got another giant isopod sitting in his burrow here, so we're just going to go momentarily and check this guy out. Oh, he might be going to come out. Oh, there's a challenge. Is he burrowing in or coming out? There we go. So this is one of these giant burrowing isopods. I think we saw one earlier on around about lunchtime. Luke was talking about it while I was down in the mess enjoying my teriyaki tofu for lunch. I think he thinks that if he can't see us, we can't see him. It's coming out.
No, my Japanese collaborator, Hiroki Kisa, just sent me back an email. I sent him a link, and he said he's interested in that epizooanthid. So, yes, it's good collection.
Ah, that's fantastic.
Thank you, team.
Yep, excellent. They look like giant cockroaches, don't they? Merrick, we're gonna start wrapping up this dive. So we're gonna start getting ready to end the dive and start recovering Sebastian. So thank you very much for joining us today, Merrick. It's really great to have extra people online that can chat to us about what we're seeing. It's very, very helpful and interesting for us, and I'm sure for everybody who's watching online as well. And hopefully we can, we'll keep in touch throughout the expedition. And when we're on some of the deeper dives on leg 2, we'll hope to be able to get you online again to chat with us during those as well. Yeah, no problem. Okay. Thank you very much. Okay.
Yeah.
See you, Merrick. So that's about it for us for the dive today, folks at home and on shore and online. We're going to wrap it up now. Thank you very much for joining us online today. It's really great to know that you're out there watching the live streams with us. We really enjoy reading your comments and having you involved in the dives with us. So we're going to sign off now. This is Schmidt Ocean Institute, ROV Falkor and ROV SuBastian, and we'll see you again on the next dive in a few more days.